Okay, so we are part of the Berberine subgroup in the new group here at ASDRP, and this is our presentation on the semisynthesis and structure-activity relationship of novel berberine analogs as DNA-binding singlet oxygen photosensitizers.
Yeah, so a lot of the research here at ASDRP really focuses on these natural products with medicinal properties and potential, some examples being Taxol, which is very well known as an anti-cancer agent, among many others. However, we're specifically interested in berberine. So to give it some background, berberine is a natural product alkaloid derived from plants in the genus Berberis, and it's known to possess a variety of medicinal properties, including antibacterial, anti-cancer, anti-diabetic, and a lot more. However, one of the interesting things about berberine is that upon photoirradiation, it sensitizes triplet oxygen to singlet oxygen, which is a highly reactive oxygen species, which can then oxidize guanine residues in DNA, those guanine bases, and then it can result in irreversible DNA damage and eventually cell death. So in our research, we really hope to focus and investigate berberine's photosensitizing abilities and optimize them.
Yeah, but before we could start our synthesis of berberine and its analogs, we wanted to know if berberine itself was indeed a good photosensitizer. So what we did is we compared it to two commercially available photosensitizers, methylene blue and rose bengal. And for the trial when we did the trial for berberine against those two commercially available photosensitizers, we found that all the photosensitizers were successful in generating singlet oxygen. However, rose bengal generated the most singlet oxygen, so there was room for improvement of berberine's photosensitizing abilities. And we'll go further detail into how we were able to find that.
So like Shelley said, we evaluated the singlet oxygen photosensitizing properties of berberine and their reduced analogs against two commercially available photosensitizers, rose bengal and methylene blue, using time-dependent density functional theory calculations. And then in order for a photosensitizer to be a singlet oxygen photosensitizer, the energy gap between the triplet excited state and the ground singlet state must be at least 0.98 electric volts. And we found that all the tested compounds were indeed photosensitizers. And we found that the reduced berberine analogs had a greater energy gap between the triplet excited state and the ground singlet state than berberine. Okay, so moving on to our experimental design. In order to conduct this experiment, we filled each of three NMR tubes with berberine, rose bengal, and methylene blue, and infused them with oxygen from an oxygen tank. We then photoirradiated them, as you can see from the pictures on the side, and then we took proton NMR of these NMR tubes with the solvent of deuterated acetone to run this experiment. And then within the singlet oxygen trapping, a 4+2 Diels-Alder-like reaction is performed, which can be seen in this top schematic, to form a scarodol, which has an endoperoxide that can be tracked via NMR, and we'll see that in some later slides. P-cymene, a minor oxidative product of alpha-terpenine, was also formed via two separate mechanisms mapped out briefly in this schematic. We integrated these peaks to yield the analysis of our experiment. Okay, and this is tracking our endoperoxide formation over time. And on the next slide, you'll see the exact places where we saw the formation of endoperoxide itself.
So now I'll be just going through four proton NMRs of some of the products that, in this synthesis. So this first one is of a scarodol, and you can see the vinyl protons of the molecule, and you can see they generated a doublet at about 6.4 parts per million. And on the next slide is one of cymene, and its aromatic protons generated a doublet of doublets at around 7.13 parts per million. And on the next slide, we have dimethyl terephthalate and its aromatic protons, and it also generated a doublet of doublet at around 8.1 parts per million. And its methoxys create a singlet at around 4 parts per million. And on the final slide, we have the proton NMR of alpha-terpenine, and its vinyl protons are highlighted with their doublet at 5.5 parts per million.
So here we have the results of our experiment. So after we test the compounds under both LED and UV light, we found that in both cases, all the compounds were able to generate single oxygen. However, as you can tell, berberine is the yellow line, and we realized that their photosensitizing properties wasn't as good as rose bengal and was only comparable with methylene blue. In addition, it only performed significantly better than our reduced analogs, which led us to believe that the serum reduction of berberine actually decreased its photosensitizing abilities. Therefore, we decided to move on to some analogs of berberine. As I briefly mentioned, I will be going over some of the analogs, and yeah, we can go from there.
Yeah, so moving on, we began, as Maher said, our semisynthesis modifications of the berberine scaffold, namely the synthesis of berberine through demethylation. And it's not on the slide, but we also did like cross couplings at the C9 position. And on the next slide, we also did like Grignard additions at the C8 position. So for the Grignard addition specifically, we used a variety of Grignard moieties, including methyl, ethyl, allo, terbutyl, and phenyl side chains. So we did this because earlier we actually did an in silico study on these analogs and evaluated carbon 8, carbon 12, carbon 13 analogs. And we found that those with allopathic substitutions at the carbon 8 bond best to G-quad DNA. So we performed all these synthesis reactions in an ice bath in tetrahydro THF, basically in good yield. And through adding such moieties to the berberine scaffold, we hypothesized that berberine's photosensitizabilities will change through modification of the electronical structure and its ability to intercalate DNA.
So we then extended our in vitro testing of berberine analogs by running Kirby-Bauer assays, which were meant to test the antibacterial activity of berberine and its analogs. So we tested berberine and its analogs in 4 concentrations, 10 millimolar, 1 millimolar, 0.1 millimolar, and 0.01 millimolar, along with testing these analogs with 2 types of bacteria, Staph E and B. cereus. And to ensure validity, 3 trials were conducted. And we hypothesized that when irradiated with UV light for a limited period of time, berberine analogs' antibacterial activity would be augmented. But after several runs, we saw that berberine and its analogs lacked antibacterial properties because we didn't see any radius of inhibition. And so as a result, we decided to move toward anti-cancer assays, or MTT assays. So our MTT assays will utilize CHO cells, or Chinese hamster ovarian cells, to test the efficacy of our analogs in obstructing cell growth, viability, and reproduction. And we'll be testing our analogs in the same concentrations that we previously mentioned for our Kirby-Bauer assays, and use these results as a means for future research regarding berberine and its effectiveness in the medicinal world. So we would like to thank our advisor, Edward New, for guiding us in this project. We would like to thank ASDRP and the Olive Children's Foundation for sponsoring our research. Thank you to Nanalysis, to Thermo Fisher for providing us with the instrumentation to conduct all of our research. And thank you all for listening to our presentation. Are there any questions?